First question, who's that fella? So I have a little bit of a wild story to tell you, and I don't necessarily know that all the facts are facts. So all the things I'm going to tell you about are facts, but I think most are. But let me say how excited I am to be here and seeing you all. It's been a while in some cases. Some of you may not know me, so I'm Jean-Philippe Gilbert. I'm an assistant professor of biology and also UB faculty. And today, you know, I'm really excited. What am I doing? Oh, okay. Thanks. Today, it's my real pleasure to introduce Christopher Kilner to you all in his public defense talk, which is really the last part of, toward the completion of his PhD. He already successfully defended privately his PhD, and today we wrap it up. And I think it all starts here. This is what Christopher looked like the day we met. I didn't take that picture myself, but I got hold of it. And I remember he came to me to ask me whether I could be part of his PhD committee, and I said, yes, of course. But I asked him, what do you work with? And he said, and I'm quoting, I work with charismatic megafauna, right? 5 years later, he's here to tell you about his work with uncharismatic microfauna. So I think it's safe to say that Christopher has been one heck of a journey. And I'm going to tell you a little bit about that journey. I just took a picture of Christopher right after that journey. That's, but you all are too smart, right, to know that that's not actually Christopher. The real question is, who's this person Christopher is with? So I'll take any hypothesis you may have. Who's this person?
I don't know. Pretty close.
Pretty close.
Pretty close.
Yeah, pretty close. So as it turns out, awesome hypothesis. This is His Excellence Michael D. Higgins, not the Prime Minister, but the President of Ireland. So what I'm going to tell you here is a testament to sort of conversations that Christopher and I used to have. You may or may not know this, but Ireland does have a president, but the president is not actually a president. That would be the Prime Minister, right? But the Prime Minister is not a president either, and it's not called Prime Minister or a president. It's called a Taoiseach, my Taoiseach. And of course, I wouldn't know this, but Christopher told me, and this is one of the many reasons why I'm going to miss our conversations. But perhaps I'm telling you this story because it embodies who Christopher is, right? Christopher is one phenomenal, greedy go-getter, right? He's always, trying to find a way to make sure he accomplishes his goals. And if that involves finding the president of Ireland, he's gonna do it. And funny enough, when he came to my lab, for reasons that will become apparent in just a second, I remember telling him, Christopher, you will need to be greedy and determined to make this happen. And today's a testament that he's been that and a lot more. And so I would say, the story starts with, this precocious young boy that from a very young age professed a profound love for tigers, sports, and sweets. I've been told that. Apparently, I don't know, Mark, what was Christopher doing that day with all that chocolate in his mouth?
We were at a Rockies baseball game, Colorado Rockies.
And what's the story about the chocolate?
Always eating it.
He still has an addiction.
There you go. Well, so I've been told that I absolutely have to say that Christopher has a profound love for chocolate and sweets. But perhaps that doesn't compare for the love he had for what Grandma Peggy, a biochemist, instilled on him, right? True love for the natural world and the scientific endeavor. And so it was through that outsized influence that Grandma Peggy had that Christopher decided, okay, a career in science is actually in the cards for me. And so as one does, he became a croc handler in Australia. The thing about truck handling that you may not be aware of is that it doesn't actually pay that much. It's also really not good for limbs. And Christopher was really attached to his. for a number of reasons, including the fact that he was a phenomenal rower in high school. And you might be picking up on something here. We're in for a wild ride, and there's a reason for it. Just bear with me for a second. He was so good at rowing that he won plenty of gold medals and awards for it, including, I've been told, the Olympic gold medal in Atlanta in '96. He may or may not have been born at the time, but he already had a gold medal. Apparently, he also did a short stint for the Durham Bulls. Here's the mascot, you know, feeding him some protein to bulk up so he can bat. I don't know anything about baseball, so all of this may be wrong. But what's actually true, this did happen. Perhaps you told me this was the most important achievement in your sports career, was his 1974 FIFA World Cup. He actually led West Germany, right, to their second World Cup after beating the most influential team in the history of soccer, you know, Johan Cruyff's Netherlands. He scored the final goal. Of course, what else is there to do after that sort of sports career? And Christopher knew it and decided, let's go back to my roots, right? Grandma Peggy, science, that's the route for me. And so he, in true Christopher, you know, fashion, he started as a student in biology and political science at the University of Scranton. This is actually true. This is Christopher as a valedictorian. You can see that chest filled with gold medals. Those are not rowing medals. These are, you know, academic accomplishments. There's more medals in that chest than I have magnets in my fridge, right? So there's a ton of them. And that's a testament to, you know, Christopher's true academic prowess. After which he would go and do a master's in Ireland. Sadly, I don't have any pictures of your time in Ireland. But
They're really rainy. So.
A lot of Guinness, perhaps. But I have it, I have good intel that tells me that at that time he picked up on rugby and actually led the Irish, you know, team to their first Six Nations victory against Scotland. Man of the match that day. After, you know, all of this, what else is there to accomplish, right? But he came back to the US and won a Tony Award. Nuts. And so here he is with his family, sister Megan, mom Maura, dad Mark, brother Matt. And so what else is there to accomplish for someone like Christopher, right? Happy kid, amazing rower, person of faith, you know, Tony awardee, croc handler, and valedictorian. And so he decided that the best thing to do was to come to Duke and do a PhD. And let me say the following thing. This is going to be a little hiatus, right? There's one thing I'm going to say, is that these 6 years have been some of the highest highs and lowest lows for Christopher, right? But that's your story to tell. So I won't. But here's the reason as to why I've been telling you this wild story. You know, if we plot the chance of successfully completing a PhD against the time, you know, somebody takes before changing labs, right? This is actual parameterized data. You know, with actual data, I can guarantee this is the actual function. You know, the first 2 years, if you change labs in the first 2 years, you're good. Beyond that, chance becomes smaller and smaller and smaller. Christopher actually changed labs and you know, came to my lab around here, right? And not only that, this was happening in the context of a global pandemic. So why have I been telling you this wild story? Because somehow he managed to make this work, right? And this is the sort of situation that it doesn't actually matter how smart you are. It's actually really hard to come off, you know, the other side of it, actually defending a PhD in the way Christopher will show you, you know, he did. And that is a testament to the phenomenal scientific prowess, but also just unabashed determination and grit Christopher has shown in these 2 years. And so, as far as I can tell, if he can do this, he might as well be a Tony Award winner and a croc handler and a World Cup winner, because this is the sort of guy that makes the impossible seem possible. I forgot about that name. So I'm wrapping up. Christopher took upon science because of the influence of Grandma Peggy, but also had, the support every step of the way of his family. And since I have you here, let me just say, and I believe this, that you all have given Christopher the deepest, most meaningful show of true love possible, which is the gift of an education. And Christopher took that to the very end in the rest of history. So take it away.
Well, I don't know how to follow that. I am truly grateful for everything that you've done, John. The research I'll show might have been the work that I did, but I wouldn't have been able to do any of it if I didn't have someone who believed in me. And so I'll always be grateful and owe this PhD to you for that. So I'm going to go ahead and get started with this public defense. As John said, I defended privately a few months ago. And today I'm going to talk about the impacts of disruptive environmental change on vital microbial ecosystems, and we'll journey through a few different systems. But first, I just want to start by going back to something that John had alluded to, which was that I had to change labs after my 4th year. And not only did I have to switch labs, but I had to start completely from scratch. And so I couldn't bring any of the work that I'd done in the prior 4 years to Jean's lab. And the first data set that went into this dissertation I got in February of 2022. And so in 15 months, this is what came out. But as I said, none of this would have been possible with Jean, but none of this also would have been possible without Danielle. And so I'm going to start the presentation off with the person who was most key to all of this work, which was you, Danielle. And so I'm so glad you could be here to see this today. I'm going to start with the acknowledgements because of the creative, like how creative and twisted this journey was. So I'm going to start off by thanking my advisor, Jean, who, if now all of you know, is deathly scared of the rain. And so he will not follow you into the rain. And my lab manager, Andrea, who was there every day, especially last December when walking into the lab was not sure if I'd make it every day. And so for her, I'm so grateful. And all of my lab mates, from Z and Kat to Richard and Melody and the +1s. And Dan is in a different picture because I got your photo late, but you're going to be coming up in the slides. Thank you all. And thank you all to my Duke community and Ecology for the sports moments and the hiking moments and the union moments and everything. You've been there for those past 6 years. And I could never have done any of this either without my family, my grandparents, and my brother and sister, and my parents, and our 3 dogs who have always been there, rooting me on, and been there for all of my graduations. And finally, I don't think any of this could have happened without my chosen family, my friends from Scranton and high school, who have been there and who taught me that's not where I breathe, but where I love, I live. And so they're the ones who, at the lowest moments of can I keep going, would call me up and say, if there's anyone who can do it, it's you. And so now we're going to go from that really joyful thank yous to talking about fires and climate change and all of these wonderful things. But the story is not completely doom and gloom. There's some positives that I found in the research. But today we're going to talk about the impacts of disruption on vital microbial ecosystems. And so we're going to start with a brief introduction of what microbial ecosystems are. We're going to talk about climate change and peatlands. We're going to talk about forest fires. We're going to talk about what is the connection between all of these, and then we'll wrap it up with a little bit more of a positive message. And so let's start with, what are microbial ecosystems? Well, for all of you Star Wars fans, they're the force that holds everything together. It might be invisible, but without it, nothing could really live. And so the microbes undergird the tree of life. They provide the balance and the force of Earth's climate through cycling all of the minerals and nutrients. They facilitate community responses to both natural and artificial disturbances. They determine succession. And they account for 20% of the global biomass, despite their diminutive size. And a majority might be in the oceans, yet terrestrial microbes, we shouldn't discount them. Terrestrial microbes are major both consumers and producers, and they have an outsized influence on climate and biodiversity, especially given that a lot of them occur in soils and peatlands that are going to be very disrupted by climate change. And so I want to talk today about microbes specifically in the context of their direct and indirect control on the carbon cycle, serving as either sources or sinks of carbon. Often within peatlands and forests, these are the largest terrestrial carbon sinks, but they can become carbon sources depending on how climate change occurs and how rapidly. And because of shifts in temperature or nutrients or resources or disruption, these sinks can become sources of carbon, whether by microbes switching their strategy from autotrophs to heterotrophs, or by fire releasing carbon. Microbes are also Earth's natural concrete, so they're key drivers of fundamental processes structuring biodiversity. They provide stability to the ecosystems, especially in successional dynamics. And they often facilitate this community metabolism where individually no microbe could process nutrients or minerals alone, but together they can process complex chemicals and create kind of this community metabolism that's necessary for higher trophic levels. And today I'm going to talk about a few different ways we measure microbes. And so I want to set you up with just this few of the different techniques that I'll be talking about. One is using DNA or RNA sequencing to understand what is present and get composition. And then the other is to use instruments to obtain traits. Because microbes are so small and difficult to study, they're not like baboons where we can study their behavior really well. And so we often have to use instruments to kind of ascertain what's occurring under the hood. So we can use chemistry or physical, optical instruments, et cetera. And so these are a few different techniques that I'll be talking about today. And for today's study, I'm going to be focusing on two specific microbes. We're going to be talking about protists, which is this larger image here. And then I'll also be talking about bacteria. But we're going to kick it off with the peatlands. And so in this first chapter of my dissertation, I explored how climate change factors, so elevated temperature and CO2, might interact to shift the microbial composition in the spruce whole ecosystem warming experiment. And so what is the spruce experiment? It's a Department of Energy experiment up in Minnesota where they put these mesocosms that are, the size of a basketball court right over the peatlands, and they construct these pipes underneath. And so they control the warming and elevated CO2 tightly in these experimental systems. And the main question they ask is, how does elevated climate change impact Northern Hemisphere forests and peatlands? And so here's what we know about forests and peatlands in this experiment. They're dominated by sagrum mosses, which sequester carbon. These peatlands store twice the amount of carbon as forests. So even though peatlands only make up 3% of the Earth's surface, they store 100% more carbon than global forests. And this carbon storage is greatly impacted by microbes, which can impact moss health or directly impact the kind of bacterial and microbial dynamics that are occurring. And so protists specifically both directly impact the carbon storage of peatlands by serving as autotrophs or indirectly by moderating the complex food webs that occur in these peat microbe systems. And so I came onto this project after a lot of the data was already collected by, here you can see Sam, a research assistant in the lab, and Jean. And so the questions that I came on to ask are, how does simulated climate change impact protist abundance and diversity using both RNA and then also the functional traits that I'll talk about. We want to ask whether temperature changes the functional traits of the community, whether it changes the composition of the community, and how are these changes related. So in the experimental design, it was multifactorial. So we had this ambient, which is plus 0 degrees centigrade with ambient CO2, up to 900 parts per million CO2, and then 5 temperature treatments. So they didn't elevate the temperature at all the way up to elevating the temperature 9 degrees centigrade over ambient. Then samples were collected from these chambers of the peat and the moss itself, FedExed overnight to the lab, where then they were filtered and incubated and collected and then sent through fluid imaging. And from there, we could obtain traits of the protist. And so then we wanted to ask, well, first, what do we expect and how are we going to analyze this? And so given the different temperature treatments and different CO2 treatments, we wanted to ask how 4 specific traits would change. And then the next slide, I'll talk about why we chose these 4 traits. It has to do with metabolism and metabolic theory. But I just want to lay out for the rest of this chapter, ambient CO2 treatments are going to always be purple. Elevated CO2 treatments are always going to be gold or brown. And then within these little protists, you can see the trait we're asking, whether it's size, shape, or what's going on inside the cell. So our hypotheses are based off of metabolic theory, where size matters. And so as organisms get larger, their metabolic rate also increases. But we also know that higher temperatures lead to higher demand on that metabolic rate. And so to meet this higher demand of metabolic rate, individual cells will decrease their body size. They'll increase their surface to volume ratio. And they might also increase kind of the machinery within the cell to meet this higher metabolic demand. And so the cellular content density should also increase. Now, we don't have a specific hypothesis on what's going to happen in resource acquisition, but we might expect that given elevated CO2, you might have shifts towards autotrophy, but at higher temperatures, heterotrophy is more efficient. And so we might expect shifts more towards heterotrophy. Now for the results, I'm going to be talking a lot about interactions. And so I just want to kind of give a refresher for those who maybe don't work with regressions a lot, what an interaction is. And so it's when you have 2 different variables that their combined effect on the dependent variable is different than their effect of each independent variable on its own. And so, for example, this is always one that I love. If you were to just consume lots of sodium on its own, you wouldn't do very well, or if you were to consume a lot of chlorine on your own. But table salt, you need that. And so visually, you can discern interaction effects on plots when the slope changes given a secondary treatment. And so you'll see lots of interactions, and I just want to lay that out before we get into 3-way interactions. And so the first question that we wanted to ask was, what happens to the overall abundance of microbes, or protists specifically, under these treatments? And so what you can see here is that under the ambient treatment, as temperature increases along the x-axis, the total abundance of the protists increases, and that's going to be on your y-axis. But then under the elevated CO2 treatment, you see the opposite pattern. And so the abundance actually decreases given that CO2 treatment. And so that's that first interaction. It's a little bit harder to see, but on panel B, this is the relative abundance of the different sizes. So these are colored by size class. And so what you can see here is that as temperature increases under elevated CO2, smaller things become less abundant and larger things become more abundant. And again, you get a reverse in that pattern under ambient CO2. And so we have this interaction with body size and temperature. And so we're going to call this functional divergence, because when we look at our traits, we get that same pattern of a switch. And so what we see is that under ambient CO2 in this purple, as temperature increases, our hypotheses are confirmed. We have things get smaller, things get less round, so they increase their surface to volume ratio, and the cellular density increases. And so our hypotheses are correct under ambient CO2 treatments, but what we see is this pattern's completely reversed under elevated CO2 treatments. And further, what we also see is this three-way interaction. So just the reversal in CO2 treatment, that's a two-way interaction. But now we have a three-way interaction because the magnitude of that change increases as you go from along that x-axis from the small size class to the largest size class. And so bigger things get bigger, or bigger things get much smaller than smaller things. And we can plot this out by doing a log-log plot. So if we just plot the log of the slope, the beta coefficient, along with the log of whatever trait we're interested in. So we have the length of the cell, which is a proxy for its size, and then the different traits we're interested in. Anything that is either super or sublinear is an indicative of an interaction. Now, what's happening with the composition of the species? So we talked about the functional composition. So how does the community's function change? But how's the community's composition change? And so using the 16S rRNA data, we could ascertain how temperature increases and CO2 treatment affect the community. And what we see is that as you go from left to right on this plot, you go from the 0-degree treatment to the 9-degree, you see those colors become more and more similar, and those boxes become less and less. And so what you're getting is a compositional convergence between the CO2 treatments as you increase temperature, and you also lose a lot of diversity as you increase temperature. And so alpha diversity is just the overall number of species present, and that decreases with temperature. Beta diversity is kind of the community diversity, and that decreases with temperature. And this occurs for both species, for both CO2 treatments. But what we want to know is how different the CO2 treatments are, and that's that Bray-Curtis distance. And that also decreases, which means the two treatments of CO2 become very similar as temperature increases. And so that's a lot that happened in that chapter, right? We were looking at composition, we were looking at function, we were talking about these different traits and three-way interactions. But what's the take-home message? The take-home message is it might be complicated to put this all together, but we can. What we can do is, how does changes in composition along with changes in function tell us about the impacts of climate change. And so in this plot here on the x-axis, we have either autotrophy, where things become greener, or they are going to serve as carbon sinks, or heterotrophy, where they serve as carbon sources. And then the biomass, so the overall abundance of those organisms. And so what we can see here is that under ambient CO2 treatments, things generally increase in biomass, but overall, greener things increase more in biomass than redder things. And so there's putatively a lot more autotrophy occurring. Now, under elevated CO2 treatments, there is a whole lot less autotrophy occurring. Everything becomes redder. Everything becomes more heterotrophic. But compared to the normalized baseline, there's a whole lot less of that happening because there's a whole lot less organisms that are becoming more heterotrophic. And so overall, there's a complicated message that, yes, as temperature increases and CO2 increases, there might be more heterotrophy, which would be net source of carbon, which would accelerate climate change. But this might be mediated by a complicated three-way interaction where with a decrease in biomass and a decrease in abundance of those organisms, there's going to be complicated carbon dynamics. And so that's something for someone else to explore in future spruce projects, but it's not a complete doom and gloom message. Forest fires, a little bit more gloomy, but we're going to talk about a natural experiment now with forest fires in the Chimney Tops Fire, which occurred in Great Smoky Mountains National Park. And we're going to ask, does fire drive changes in microbial diversity? Here's what we know about the effects of forest fire on microbial diversity and forests themselves. So we know that wildfire structures ecosystems. Fires is one main component of the global carbon cycle, but a lot of whether or not carbon is released or even absorbed post-fire is dependent on the response of the microbial community. There's not a lot of plants left active after a major forest fire, but a lot of soils can still serve as carbon sinks. But most of the studies that have been conducted have occurred in chaparral or pine ecosystems, which are a lot more fire frequent, but that doesn't mean that, especially as climate change occurs, less fire-dominated systems might become more fire-dominated. And so we want to explore what happens in these systems. And so we want to know, does fire drive consistent change in soil bacterial diversity? I also want to ask if there's a phylogenetic signal to this pattern of change. And whether or not the microbial community returns to its baseline quickly after a disturbance event, especially given that this occurred in a fire infrequent ecosystem. And so again, this fire occurred in Great Smoky Mountains National Park. I'm calling it a natural experiment because I didn't go out and set the fire and design the experiment. A couple teenagers did that for me. So the Chimney Tops 2 fire occurred in 2016 after some teenagers set a fire near Gatlinburg, Tennessee. And it was right by National Ecological Observatory Network sites, or NEON sites. And so they had already gone out in 2016 and collected soil 3 times. And they had already planned to go out in 2017 and collect soil. And so now I had pre-fire and post-fire soil data. But not only that, this pre and post fire was separated between burned and unburned sites. And so most prior fire studies either burned everything and they had pre and post, or they only had burned and unburned. But this allowed me to have both pre and post burned and unburned data. And so I could ask a little bit more in-depth questions about the effects of fire on microbial diversity. And the results were not entirely unsurprising. Bacteria do not enjoy fire. And so what you can see in the data here, pre-fire is in blue, post is in orange. There's no statistical difference in the alpha diversity in panels A and, well, in panel A or beta diversity, which is that community diversity in panel B, in the unburned sites. However, in the burned sites before the fire and after the fire, there's a significant reduction in both the alpha and beta diversity. And then there's no significant recovery in either the alpha or beta diversity in those burned sites over time. And so the C and D panels are only in the burned sites. And we were able to look at 3, 6, and 9 months post-burn. And there doesn't seem to be any significant recovery in the diversity. And so there's major reductions in the total ASV richness, as I said, and then there's also reductions in the community diversity. And so that led me to ask, okay, well, what is driving these changes and where are these changes occurring? And from there, we can look at specific phyla of bacteria and ask, where are the biggest differences occurring? And so there's clear difference in the phyla in the abundance and composition. So unburned is under the blue, burned under the orange again, and pre and post are next to each other. Now we can see there's greater turnover and variability in those burn plots. The patterns are, a few sites actually increase post-fire in their richness. Most of the sites decrease, and kind of the variability in those colors that you see, shows that there's a lot of variation across these different sites in which phyla respond most to the fires. So then we want to look even more in depth to see, okay, how are these specific phyla changing? It turns out some bacteria do really like it hot though. And so in this table, the bolded bacteria are the ones that change significantly pre and post fire. And you can see a few changed a lot. Cyanobacteria increased 194% in the burned plots. So after burn, cyanobacteria take off. Fibrobacteriota, they absolutely take off, 238% increase. A few different phyla appear for the first time after a fire. But then you have other ones decreased by 91%, 80%. And so when we want to scale it up even more to these kind of superphyla that are around more of the functions of the bacteria, we can see one really took off and one really decreased. And so what's going on there? CMS, that superphyla holds a lot of photosynthetic bacteria. And so it seems after the fire, whether from light availability because of increased light attenuation through the canopy, or kind of new nutrients and minerals available after the burn, photosynthetic bacteria did really well. Whereas bacteria that were gram negative did pretty poorly. And so, there's a lot of variation in what's happening. So then I wanted to ask, well, if there's these kind of clustering in the superphyla of bacteria that do well or don't after a fire, is there a phylogenetic signal? And the answer is boring, it's no. So running a chi-square test after developing a phylogeny, there's nothing significant in the differences of whether a species responded positively. So this is an indicator species analysis. So positive is in purple, negative is in brown. You can see that I really think about the Roman Empire a lot because I use their colors. And so, in the chi-square test of indicator species, there's no significant relationship phylogenetically. Let's go a little bit more in depth and use some pretty powerful phylogenetic distance scores based off this phylogenetic tree. And there's no response. The distribution of this plot doesn't matter necessarily, but if the blue and the red distributions, if their means were within kind of the 95%, there would be a significant response, but there's not. The distributions are very far apart, and so the phylogenetic score is low, and there doesn't appear to be a phylogenetic signal in how the species respond either positively or negatively to fire. So then what drives the overall consistent reduction in alpha diversity and beta diversity? We're kind of constrained by the site. And so we're limited by the NEON data availability in terms of total environmental factors. But what we do know is that where the soil was sampled, that's the biggest driver of kind of the results. And so in this first plot in panel B, this is a kind of a mean distance score of the PCA results, principal component analysis results. But what you can see here is there's this weird V distribution that seems to be along this first axis. But if you look at the actual individual sites, they're very clustered near each other, whether it's pre-burn in blue or post-burn in orange. And so that led me to ask, okay, well, if site's the biggest driver, where does the environmental effects like fire or soil pH or moisture come in? So in the second panel C, I did a redundancy analysis where I say, okay, we're going to take that axis of site and set that essentially to 0. So we're going to take up all of the site information, and then we're going to look just at the environment information. And from that, we can see that fire is significant in driving the community change. And we can see that soil pH is also very significant. Now, we might expect that soil pH will change because of fire, so there's some collinearity there. But pH and fire are both very important. And what's a cool pattern is that the seasonality resembles a fire and pH. And so as the seasons progress from spring to summer to fall, so that +3, +6, and +9 months, the baseline kind of returned from the burned plots in orange to the pre-burn in blue. So you can see that fall eigenvector is most similar to the pre-burn. And so, I don't have the plots here, and it was, extra analysis, but essentially the take-home message is, if you dive even deeper into that, there's a lot of turnover that starts occurring as time moves from 3 to 6 to 9 months. So there does appear to be some start to recovery, but a lot of other bacteria start popping out. And so you have lots of turnover. And so the overall richness does not increase, but the composition turns over a lot. Okay, and so now I'm going to shift focus again. So we start with peatlands, then forest fires. Now we're going to just talk about the individual organisms themselves, and we're going to talk about body size. As we saw in the first chapter, body size was an interaction. And so I want to investigate more the effects of body size on response to the environment and how environment affects body size. And so this is going to be a lab-based experiment. We know that body size plays a fundamental role. We already saw that in the first chapter. And my lab, Jean's lab, had worked a lot already on body size. So Dan has this wonderful paper where he looks at how body size in different species might influence their response to temperatures and their growth rates, their metabolism, their reproduction. And so overall, body size is really important. We also know from one of Jean's older papers that body size can affect trophic position of an organism. And that can also kind of restructure food webs and affect how energy flows across trophic levels. And so overall, body size is really important. And we know that in mammals, it can also affect behavior. And so we developed a few research questions. Specifically, we want to know how temperature, genetic diversity, resource availability, competition, so this is really small factors that might affect things, impact body size. And so I want to ask specifically how these impact body size, but how these impact density as well. Because earlier we saw that body size seemed to correlate with the overall abundance of species in that spruce experiment. And we want to try to put this all together in a unified model. As you can see, there's kind of lots of really cool math, and it was really great to take the opportunity to learn under Jean a lot of this theory and kind of rekindle my interest in math. And so for these kind kind of this unified model, I'm going to rely on 3 different equations. I'm not going to go too in depth in the derivation, but the lack of Volterra, this essentially is going to be how does competition and intrinsic growth affect the total number of organisms, or the density. And so R is the intrinsic growth rate, K is carrying capacity, N is the total number of the organisms. That small m is a mortality term. That alpha there is going to be competition. Don't worry, you don't have to remember these. There won't be a quiz. Then I'm going to take an equation from one of Jean's papers last year, and I'll explain the components a little bit more on the next slide. But this has to do with kind of resource economics, essentially. And then connecting that intrinsic growth rate and carrying capacity to the actual body size of an individual through these kind of scaling relationships. And so this is the derived unified model. And so this first component is the change in body mass. That popped in really quick. The second component here, that's going to be the phenotypic plasticity. And so that's how an individual itself can respond to a changing environment. This third component here, that's the genetic heritability. And so how kind of responses might be carried through generations. And then finally, this component here, this is the change in fitness. So this is whether the response is a net positive or a net negative for an organism's ability to essentially maintain metabolism, reproduce more. Essentially, it's the fitness of the organism. So it's a complicated model, but because of great lab mates who assisted in this experiment, we were able to do this in kind of a great effort. And so we wanted to change temperature, we wanted to change nutrients, we wanted to add in competition, and we wanted to change genetic diversity. So we set up this multifactorial system. Again, we incubated, we used fluid imaging, and then we tested our hypotheses against the empirical data. And so kind of this mind-melding math runs into what occurs in empirical experiments. But we expect, again, based off that temperature size rule, for temperature to lead to a decrease in body size and an increase in the density of organisms. We expect that lower nutrients will lead to a decrease in body size and potentially an increase in density. Competition to decrease body size and density, and for the genetic diversity to increase both body size and density. And so these are based off of kind of what already exists in the literature, or what we might expect from simulations of our model. Our results are that size matters a lot. And so higher temperatures do decrease body size. And to orient you on this kind of complicated graph, on your x-axis is days. So we sampled 3 time points, 1 day after incubation, 7 and 14 days later. A through D is going to be body size, and E through H is going to be density by these different treatments. So higher temperature decreases body size, higher genetic diversity increases body size, less nutrients decreases body size, and competition also decreases body size. So all of this is kind of what we expected from our hypotheses. And then we do see that inverse in a lot of these relationships between density and size, where higher temperature led to higher density, or higher genetic diversity at some point led to higher density, but actually lower density in panel F by day 14. So that's the only one that broke kind of the pattern of our hypotheses. And then we want to fit our empirical results to our model and derive from that the kind of coefficients for the different components of our model to understand what's really going on underneath the hood. And from this, we found that there's interactive effects of treatment and time for the temperature treatment, for higher genetic diversity, for less nutrients. And so overall, the effect of these individual treatments increased over time. Which we might expect. These organisms are responding both plastically with the phenotype that they can, and then they might, in reproduction, be responding genetically as well with higher genetic diversity. And so we could expect that these treatments might have a higher effect over time. Now, what was the most fun for me was then taking our model. So we had those empirical data on the last slide, then we fit our model to it, which is those smooth lines. And from that, we could get parameter estimates. And then using those parameter estimates, we could kind of create simulations and plot change in body size, which is DMDT, versus the starting body size at time point 1. And then at time point 2, we want to look at the change in body size. And we can do that for density and then over time as well. So we can look at the effects of body size itself, density, or day on the change in body size. And what we see are these really cool oscillation patterns where, as density approaches carrying capacity, the change in body size approaches 0. And so as things get close to their kind of carrying capacity, there's very little change in body size. But as body size increases, there's a lot of change in body size. And so we kind of recover that same pattern that we saw in chapter 1, where bigger things change more than smaller things. And so, there's some really cool patterns that came out in these simulations from the parameter. And then overall, we also found that as time increases, the variation in change in body size also decreases. And so those oscillations kind of peter away. And so overall, while this experiment, there was a lot of messy data in how body size might change with these different treatments of higher genetic diversity, increased competition, more or less nutrients and higher temperatures, we recover consistent trends in the model itself. And so to me, that is a clear indicator that the model's onto something, and there's a lot more kind of experiments to be done. I know I'm starting to run out of time, so I'm going to quickly just talk about these parameters. And so, to kind of hone in on the hypotheses, the parameters ended up aligning with theory. And so that temperature drove the greatest change in plasticity. Competition drove the greatest change in kind of that lack of altera component. Nutrients affected body size the most in terms of the resource, a kind of economics model. And then genetic diversity obviously had the greatest impact on heritability. I don't know why that popped out really quickly, but we'll replay it for everyone again. But the point is that what we expected is kind of what we saw. And so that shows that there's a lot of good grounding in that theoretical model development, but a lot more to explore in terms of some of those oscillatory patterns or those interactions. And so now I want to get to conclusion, and I'm going to try to sum it up pretty quickly because, like my PhD, which was a sprint, I know that this presentation was a lot of information to also kind of sprint through. And so in conclusion, the main takeaways from chapter 1 is that there's a 3-way interaction between temperature, CO2, and body size, and that this is really important for functional traits in protist microbes that might have a large impact on the global carbon cycle. In chapter 2, we found that fire reduces the diversity, both alpha and beta diversity of a soil microbiome, and shifts the dominant groups and the major players, and that there might be some compositional turnover that leads to kind of minor recovery over time. And then in chapter 3, we find that the environment intimately shapes body size dynamics, so how body size changes over time, and how that change in body size affects the density and population of the protists. And now I want to spend a little bit of time talking about the future. So I defended privately in July, and then in August, I started at the Bird Conservancy of the Rockies, where I'm now applying that theory and empiricism to conservation challenges. And so last week was kind of an exciting another sprint that I think my PhD prepared me very well. The CEO was like, over the weekend, I need you to build this shiny web app in 5 days. And then I'm going to go present to some potential funders. And so going ahead and building this in a few days, but really applying lots of what I learned here at Duke. So applying lots of the coding that I learned here at Duke and how to structure and talk about narratives and information and how to really think about conservation challenges. Helped me put this tool together in a few days where we tracked bird movement using telemetry data and kind of pulled that migration information in, allowing these kind of users to quickly explore species of concern and kind of where they're occurring in the Great Plains. And so none of this would have been possible without obviously finishing my PhD, but really without learning a whole set of skills across my time here at Duke, from GIS and remote sensing to theory and kind of lots of heavy coding and how to think about models to kind of put all of this information together in a week for my new employer, where I get to do really great conservation work. And so with that, I just want to circle back one last time to those who made this possible. So I want to again thank Danielle, and then Kathleen, Justin, and Zachary, who were the DGSs during my time here. And my committee, Jean, Jim Heffernan, Reedus, Ariane, and Andrew. In my 6 years here, I had a variety of funding resources, from NEON to the Department of Energy, the W.D. Billings Fellowship here at Duke, ESA, Nick School, graduate school, and then a variety of different Duke projects that help me bridge semesters. And then I think it's really important to acknowledge the members of the Duke community, who I think sometimes we don't think about as being such large components of our academic journey. But if it weren't for the really friendly staff at BU Blue, where I would get coffee and they'd ask how switching labs was going, or what my new lab was like, or at Il Forno and Cezanne, where they'd sometimes, for reasons I still don't know, give me free food and ask me kind of how my third chapter was coming along. They brightened my day, and so they were incredible. And the facility staff and the buildings I've been in, who will pop in when I was still here at 8 o'clock at night and encourage me, they were all wonderful. And I don't think they get enough recognition for what we do as students here. And then at Duke as a whole, from student health and CAPS to the president's office and the library and the Catholic Center, for kind of rounding out me as a person and providing those resources, I think they played a large component in my dissertation as well. And so with that, with the few minutes we have, I'll take questions. Yeah, go.
Fire study, do you know anything about prior to fire, like these fires? Communities might lead to a bigger effect of fire on microbial community than areas that sort of right.
Yeah, so the question was, if people on Zoom didn't hear, is this a fire suppressed community in the Great Smoky Mountains, and would that have maybe led to a greater effect on the microbes in terms of the community and recovery? So prior to kind of the Yellowstone fires in the 80s, there was much more of that kind of fire suppression practice in national parks. But in terms of Great Smokies, there wasn't as much, to my knowledge, in that area. This was the largest fire that they estimate occurred in 500 years or so in that kind of section of the park. And so it definitely was a larger fire than might normally occur, given that they estimate it's the largest in 500 years. There was some really cool information on the fire intensity, and that intensity did vary between sites. I didn't look into the intensity specifically for this chapter, both for time constraints, and then also, you know, there's going to be additional information that becomes available this year on actually 3 years post-fire. And so I think there could be some really cool analysis of intensity in 3 years post-fire. But that intensity question is a great one, because I do think intensity of the fire could impact the community.
Add on to that, I was thinking about the loss of the forest floor with the fire, and then interestingly how the community was going back in the fall, which may also coincide with the fall, and that resource coming back.
Yeah, I think that. Yeah, I think that seasonal recovery was really, really interesting. I also think that litter burning up all of that kind of top layer might be why the photosynthetic bacteria recovered or actually took off a lot more than they did pre-fire. And so I think there's a lot of dynamics that could be really interesting. NEON also provides ITS data. So looking at the fungi could be, you know, a future study as well. There's just a trove of information that NEON has that I think could still be accessed and questions asked. Yeah, Dan.
Do you have any speculations about why temperature effects are reversing or elevated CO2?
So, oh, this is a great question. It's definitely something that I've thought a lot about. This is kind of almost going back to my molecular biology days, but I think that there's a lot probably occurring in terms of gas dynamics, metabolism, and how cells metabolize as to why there would be this inverse change. I think it's hard to grapple with because, you know, ambient already is almost or around 400 ppm, which, you know, 150 years ago was only 270. So there's obviously already been change that's occurred. That elevated CO2 is 900 ppm, which is, you know, a lot to think about. But I think that if I had to speculate, I think it would come down to how that elevated CO2 is shifting the kind of metabolic pathways within the cells. And that is shifting the proteins that are being used, shifting a lot in terms of the cellular dynamics. And that's going to obviously change a cell's response to temperature.